Do we need your microphone?
Brilliant.
Let's just be able to walk around.
Yeah, if I do walk around and do it all, then I'll need it. But yeah, so basically today I'm going to talk about large-scale analysis of epigenetic data, FORGEdb and FORGE2. And let's see. So I think GWAS needs no introduction here. So obviously, people who want to work on GWAS, genome association studies have been for many years now already associating common sequence variants to disease. And the focus of my research is, so you have a case control or another type of design, and then, so when you do a GWAS, you get together a group of cases and controls typically, and then you run them on a SNP array, typically from a biotechnological company, and then you identify the genomic variants that are associated with case control status or quantitative trait. And the focus of my research is, okay, so when you've done all this, how do you interpret your results? So, okay, being successful, you've done the GWAS, published, we have the summary statistics, we have all the information. Summary statistics, you've done the GWAS, you've obtained all of your information from the GWAS, and you have a list of SNPs effectively associated with case control or quantitative trait. And so interpreting GWAS results is remarkably challenging. I don't think this group here needs any detail on how challenging that is. It's rather difficult. And I'm going to give a few examples of particular variants, but it's a big challenge for the field. And so the focus of my research is trying to illuminate, and trying to effectively aid the interpretation of GWAS variants. I don't think we have a magic solution to interpret every GWAS variant, but what we're going to try to do is to improve variant interpretation. And so currently it's very difficult to assign a gene for many GWAS SNPs. Many GWAS SNPs are intergenic. So a gene-based approach just simply doesn't really work for most GWAS genes. If you just link it to the closest gene, it's just not really an optimal solution. I think this was a solution very early on in GWAS. There was a sort of tendency to sort of annotate things to the closest gene. And you can still do it, but in a large percentage of cases, and you can actually quantify that, closest gene is not that helpful. So I was faced with this problem. So what can we use to annotate GWAS SNPs? We have no closest gene information. At the very beginning, when I was starting to do my research, there was very, very little qTL data. We didn't have that much protein qTL data either. And so, well, thankfully, there's a solution. So Superman in the form of chromatin data. So I think that chromatin data are extremely useful for interpreting GWAS variants, and this isn't just a personal opinion of mine. So chromatin data, as you know, are tissue specific. They've been mapped across many different consortia. A lot of money, a lot of NIH money has gone into mapping chromatin data. And the good thing about chromatin data is that it's genome-wide. So it provides annotations for every SNP. So in a tissue specific context, it may help indicate potential target genes, and it provides information for every single SNP, be it intergenic, be it more close to a gene, be it in an intron. So one of the methods I developed was FORGE2, and so FORGE2 effectively takes GWAS summary statistic information and then integrates it with epigenomic data. And then from FORGE2, we can effectively find out which is the key target cell type or tissue of interest for a particular GWAS. So what we're trying to do is trying to identify what could be a potential cell line or target tissue to analyze, to use for analysis, functional analysis of GWAS. And so there's a large number of different chromatin data sets, and I'm sure you're all familiar with histone marks, which are typically analyzed as broad peaks in terms of GWAS data, accessible chromatins, so for example, ATAC-seq or DNA-seq. And we obviously have DNA methylation and nucleosome positioning, and not all of these data sets are equally informative for GWAS. So some data sets are more informative than others. And just to give a little bit of context here, so we have this nucleosome chromatin, and then accessible chromatin is really where a lot of the important things happen in the cell. So regulatory elements, typically bound by transcription factors, and potentially forming enhancers or locus control regions or other kinds of regulatory regions. These can be mapped by chromatin accessibility. And one of the most important data sets for interpreting GWAS is chromatin accessibility. Why? Well, because every single regulatory element needs to have something binding to it. So regulatory elements need to have transcription factors and other proteins bind it. And so when they bind, they need to bind accessible chromatin. And so that accessible chromatin is a sine qua non. It's a proxy for every single kind of regulatory element. If it's an insulator, it will be accessible. If it's an enhancer, it will be accessible. Every single kind of regulatory element has to be accessible if it's bound by transcription factors. And so we have massive amounts of chromatin data, massive amounts of different data sets, and how can we sort of bring them all together? So, as I said, DNAs have sensitive sites and accessible chromatin. When integrated with GWAS SNPs, very good at pinpointing target cell types. And so, accessible chromatin, especially H3K4 monomethylation. So, let's not get lost in the terminology here. How many people here know what H3K4 monomethylation does? How many people don't know what it does? Okay. So H3K4, so what does H3K4 mean? So histone 3, there's 4 main histones. Okay, K4, so that's lysine 4, and there's a monomethylation on the branch of lysine 4. So it's a monomethylation on a particular amino acid, which is on a histone. Okay. So you could say, well, why are you going to this sort of very specific amino acid that's on this histone? I mean, you could say, well, who cares? There's a lot of amino acids on a lot of histones. And so H3K4 monomethylation has always been associated with enhancers. It's one of the best predictors of enhancer regions. So when you have this particular monomethylation at K4, that's very useful. And it's very tissue specific, and it's a very good enhancer. And so we can analyze and integrate huge amounts of data from enhancers and GWAS, and here you have a very big graph that the Roadmap Consortium made. And on the x-axis, I don't know if you can see it, it's meant to be so, it's one of these graphs that's so big that it's barely visible. And I'm not entirely sure how helpful it is, but on the x-axis, you can see that there's a lot of cell types. And on the y-axis, you can see a lot of diseases and traits. And so you can see that these red dots are basically pinpointing the target cell type statistically for every single GWAS. So you can do this systematically across many different GWAS. You can identify the target tissue that has the highest overrepresentation in enhancers. Okay, and this is useful when you're doing a first step and you're saying, okay, I want to analyze this GWAS and I want to find the good tissue of the target cell type. And so that's what you do at the beginning, to sort of identify what is going to be your most productive cell line to work with. And so FORGE2 does all of these kinds of analyses. It expands to other types of epigenetic data. And the key in FORGE2 is using a proper statistical background. So the only way you can get this tissue-specific enrichment is when you use the right model, which accounts for SNP bias, promoter bias, etc. And FORGE2 is perfectly complementary to FUMA. I'm sure you're all familiar with FUMA. It's a widely used method for GWAS, post-GWAS analysis. And FORGE2 uses other types of data which are complementary to what FUMA does. And so, so effectively, I'm going to sort of briefly outline the statistics of FORGE2. So FORGE2 takes data from published GWAS, and it uses SNP array information to create a match background. So transcription start site, minor allele frequency, and GC content are effectively used in the model to adjust for SNP bias. And then it overlaps it against overlaps both background sets and input sets against regulatory elements, and you end up with a p-value. Okay. So that's the interesting fact about FORGE2. And you can end up with a list of different minus log 10 p-value sort of numbers, and you end up with enrichments typically for target cell types. So CD14 monocytes are enriched for this particular GWAS. So far so good, and that's FORGE2. But let's talk about the key here, the key method that we want to talk, which is derivative FORGE2, which focuses on analyzing particular variants. And I'm sure this is what will be most useful here. So FORGeDb is the next iteration to try to annotate all of these data sets. And as you know, there's a lot of epigenetic data and other genetic data out there. So EQTLs, transcription factor specific binding data sets like Cato. And you have chromatin looping information. You have DNAs have sensitive sites, you have chip-seq data, chromatin states, CAD scores, and motifs. You have all kinds of data. And so one of the biggest problems that I'm sure you're faced with is you've trying to analyze GWAS SNPs, and you're trying to find some functional genomic information, you're trying to find some functional role. And you have all of these different data sets, and it's overwhelming. To my mind, it's overwhelming. And we need a multi-omics method that really integrates these different data sets. So what FORGEdb is trying to do is it's trying to bring together all of the major annotations into one cohesive framework and to give every SNP one cohesive score that takes into account these different data sets. And so I'm going to walk you through what each data set means. So what does each annotation mean? So the first annotations that we have are closest gene. Okay, so why? Well, because closest gene, even though it's not useful for every SNP, it can give you the first indication of what does this SNP do. Okay, and so closest gene is not always the right gene, but it's good to know what the closest gene is for a particular locus. I'm sure, Ludmila, you will have seen that closest gene is sometimes the most important gene. And not always, but sometimes, right? And so it's important to know what the closest gene is and what the context is. So this is why we provide browser information and closest gene information. So I'm going to give you an example of a variant where the closest gene is actually very important. So there's a COVID-19 associated variant, and I mean, feel free to chip in because there's been an COVID-19 as part of CRL. But this particular variant was found to be associated with ACE2 expression, and it's very close. I'm sure you can see it here. It's very close to the ACE2 promoter, maybe even overlapping some segments, some of the transcripts. And so this particular variant is associated with ACE2 expression. Is important for ACE2, and so it's the closest gene effectively. The consensus in the field, or at least the major consensus in the literature, is that this is an ACE2 regulating SNP. Okay, and the SNP is here where the vertical blue line. So this is obviously it's difficult to read a SNP from this far out. This vertical, can you all see the vertical blue line? This light blue line. And the ACE2 gene starts here in black. So effectively it's very, very close to ACE2. Let's talk about a variant where the closest gene is not important. So this particular variant, RS1421085, so this variant is a famous obesity-associated variant. It's located within an intron of FTO. Okay, and it's actually one of the major associations for obesity. So this variant is very, very important for understanding obesity predisposition, at least as far as common variant can. Obviously, obesity is a complex multifactorial disease. This variant is one of the most important ones. And it's located in the intron of FTO, and people have constantly done studies on FTO. But recently, there was a really compelling study that showed it had long-range interactions with IRX3 and IRX5, especially through 4C and other high-C-related techniques. And so these genes are very far away. IRX3 here is looped in red. IRX5 is looped here. These genes are effectively, more than a megabase away, right? Or at least, in the region for IRX5. And so, the closest gene here is completely useless almost. The closest gene is not helpful here. Here we really need to know three-dimensional genomic context, right? And so this is why it's so important to functionally validate these SNPs. Okay, so I come from the computational field, but it's very important to understand these SNPs in the cellular context and function. Because if we don't have good functional experiments, we do not know anything about what these SNPs do. We can make the best models, but how can I put it? There's nothing like the real experimental data. So closest gene is not always helpful. This is important to take into account. So how can we know if it's a closest gene or not? So you can say, all right, Charles, this is great. You're telling me sometimes it's closest gene, sometimes it isn't. how do we know? So we know through functional validation, but we can also get some information using epigenetic annotations, right? And so what does each annotation mean? So the next annotation that we have for FORGEdb is a CAD score. So the first thing you want to know about a SNP, if you're computational, is you want to know how deleterious it is. So some SNPs, when edited, when altered, have almost no effect on protein expression, have almost no effect on RNA-seq, and have almost no effect on survival. And so here, the CAD score is useful because it's an aggregated score across many, many studies, and effectively it tells you how important that SNP's deletion would be in comparison with all the other SNPs in the human genome. So some SNPs some substitutions and SNPs are known to be incompatible. Some of them are incompatible with life, right? They're not SNPs, but some substitutions which you can do in some genes. If you delete certain genes, you will effectively not have an individual which will survive, right? And so obviously we're not in those extremes for these SNPs because these are SNPs in the population, so they're very clearly compatible with life. But some of them are more deleterious than others. So CAD score will tell you how deleterious a particular SNP is. And this will tell you what a good candidate will be. So SNPs, if you have 3 SNPs, which are your candidate variants, the one with the highest CAD score is likely going to be potentially a very good candidate. So that's what CAD scores are for. EQTLs. So how many of you know what an EQTL is? You know what EQTLs are, right? You work with EQTLs all the time. And so EQTLs are useful once you've done the CAD score analysis, or you've seen what the CAD score is. FORGEdb will give you the CAD score for every SNP. This EQTL is an estimate of the association, of course, of the different alleles with RNA expression. And so we included 2 different EQTL consortia. So GTEx, are you all familiar with GTEx? Okay, everybody loves GTEx. I like GTEx a lot. GTEx has some very good data, but it doesn't have a lot of blood data. So this is why we also include EQTLGen, which is another consortium, which has more blood data. So we're trying to include multiple data sets to be trying to be sort of equal across tissues, trying to have cross-tissue information. And so, as you know, there's 6 EQTLs where a local SNP, where SNP has an effect on a local gene, and then you have trans-EQTLs where there's an effect on a distant gene. And so the real difference is whether there's a local chromatin interaction or promoter gene expression effects. So this is a direct relationship of this SNP with this gene. You can see this in the correlation with expression. And then the trans-EQTLs are mostly driven by altered protein or transcription factor effects, which are affecting genes which are very far. So they may be in another chromosome. You have, for example, if this gene is a transcription factor, then any SNP that affects it will affect also all of the downstream genes of that transcription factor. So that's a trans-EQTL. And so EQTL information is good, but you may have cis or trans-EQTL, so we need to have more layers, okay, to really know what a particular SNP is doing. And so this is why we added ABC interaction data. So ABC, we have to think of ABC as a little bit like high-C data. So it's basically high-C data that's being merged with ChIP-seq data for signal effect. So for signal intensity of ChIP-seq. So why do we integrate high-C data with ChIP-seq? So this provides a better predictor. Just using raw high-C data is not that informative because you have a lot of contacts which are across the genome. And so you want to use ABC data, which kind of integrates local ChIP-seq signal and will give you a better metric of looping, of real looping of that SNP with its target. And so,
do you provide sources of your data?
Definitely. So, yeah. So I'm going to do something which is dangerous. I'm going to do a live web demonstration at the end. Okay. And you'll see that we have all of the source data sets linked. If you want to do your own analysis, that's definitely available. Yeah. And so ABC interaction is very useful. You have high-C data and you have local ChIP-seq signal on this. It's a very good predictor of targeting. It's much better than process gene. And so looping, what do I mean by looping? So 3-dimensional looping, typically enhancer promoter, but it can be other types of effects. For example, insulators can sometimes inhibit looping. So it's not always enhancer promoter, but there is a lot of enhancer promoter looping in these ABC interactions. Okay, so let's go to the next step. So motifs. So transcription factor motifs. So once you have a SNP and you know that it's an EQTL for a target gene and it's also looping to that target gene, so it's an enhancer, for example, that loops to the target gene, you may be interested in knowing what transcription factor is binding this. How is this being regulated, right? How is the fundamental biology happening? And so that's why we had transcription factor motifs and Cato scores. So a motif match, for example, in a particular SNP site, is basically a transcription factor binding model for that particular site, a match of the model. So, for example, if you have a motif match for example, MZF1 in a particular SNP, then that will tell you that you have a likelihood of MZF1 really binding that location. And then a Cato score is just a measure of allele-specific transcription factor binding from DNA-seq. So a lot of these people did these large DNA-seq experiments in Seattle. I'm actually from the team that sort of works on DNA-seq, so we're like the one team or the one, two teams that does DNA-seq. But so the team that does Cato scores were developed as a measure, for example, transcription factor binding within accessible regions. And so these two scores come together, and they're really helpful to sort of predict what transcription factor might be binding your particular SNP. So if it's an enhancer and if the SNP is disrupting transcription factor binding, you want to know what motif and what Cato score is sort of supporting that. Yeah, which will give you maybe information to do an experiment on that particular transcription, for example, ChIP-seq. And so we can predict transcription factor binding using DNA-seq because transcription factors leave grooves in the DNA-seq data. So think of DNA-seq as a mark that detects accessible chromatin. So the DNase enzyme, or TAC-seq is very, very similar. Here's an example of TAC-seq. These enzymes can only attack the DNA. I mean, a TAC-seq in a way, for Greenleaf, when he developed a TAC-seq, like it's got to do, it's not pronounced a TAC-seq, it's pronounced a TAC-seq because the enzyme attacks the DNA. Okay, this is important to take into account. So you have all these nucleosomes, and then when the DNA is open, then the enzyme can go in. So this maps open DNA. Okay, but you could say, okay, well, the DNA is open, but actually have a transcription factor that's already in that open DNA. And so that transcription factor also comes up in the experiment. So you will have an increase in signal and then a drop, because the enzyme can't get to the little fragment of DNA that transcription factor is binding. So these techniques, when you do deep sequencing, are so precise that you can actually see what transcription factors are binding within your accessible region. So you have this sort of barcode-like signal. Can you see where the region is broadly accessible? Down here, it's quite small, but I'm trying to show this at the bottom of the graph. But it has little valleys in this sort of peak. And those values are transcription factors, which are leaving their own footprint. So this is very useful. So you can get some information on transcription factor binding in an enhancer. And transcription factors are, of course, crucial because they mediate a lot of this three-dimensional looping that I showed before. And they can also mediate the insulation. So this is very important. Transcription factors are obviously key here. And so what do all these final annotations mean at the bottom? So we have DNase hotspots, we have H3 histone marks, these are just more histone marks. So then we have chromatin states. So at the end of this section of FORGEdb, what it's trying to show you is what are the valuable tissues for each particular SNP. So this time it's not like FORGE2 where you're analyzing an entire GWAS. Here we're saying for this particular SNP, what's the signal like in every cell type? And how can we know which are the best cell types? And so this is what DNA-seq, which evaluates chromatin accessibility, and ChIP-seq, which evaluates, as you know, binding or histone modifications. And chromatin states effectively just promote enhancers. These are like allocations of states at the end. So let's look at a particular example. Do you have any questions? Okay, any questions so far on ChIP-seq, DNA-seq? There's a lot of seqs, but yeah, you're all familiar with ChIP-seq and EQTLs, so yeah.
I have a question.
Yeah, go ahead.
Like in terms of the data sets, how do you factor in the tissue type and the variability that a lot of cell types contribute from a specific tissue. So, does that influence the model somehow, and how do you adjust for that if it's the case?
So these are processed data sets. So they've all been processed taking into account other tissues. In fact, that certain tissues, for example, are very difficult to analyze. So I think that's what you're referring to. So, for example, neutrophils were almost impossible to do DNA-seq on. I don't know if anybody ever managed to do DNA-seq on neutrophils, but I think they sort of auto-detonate every time somebody tries to do it. So there are some cell types that are very problematic. So what we tried to do was go to one particular consortium which had unified data and which had really compared data, consolidated data. We use the word consolidated here, it's really from roadmap, but really consolidated the data sets across tissues to make sure that tissue-specific effects aren't, they're not really technical effects that are driving this, it's all biological effects. So this is kind of one of the best data sets in terms of how homogenized they managed to make it. So it's basically, they managed to make sure that the real effects aren't driven by cell composition, for example, which I think is what you're also referring to, and they're not driven by, for example, batch effects to do with particular tissues. They're all driven by real sequence. So roadmap has been validated quite extensively, not least with GWAS data, and this is really very important to get right because we don't want to have single particular tissue just because it has one more homogeneous cell line or cell type. Sorry, one particular cell type in that tissue is driving everything else. Effectively, some tissues are very dangerous because you can get peaks everywhere, particular tissues. And so this is why it's important to have a consolidated data set that really sort of got rid of all these different effects. Yeah, it's very important because there's a lot of data out there and not all of it is of equal quality, as you know. So we can't, we have to be discriminate the best data sets, really. And
Can we, before you go, how did you write this score of 9 on the previous line? Can you explain that?
Yeah, I will explain that. So this is actually here in a specific example. Yeah, so the score of 9 is derived. So for this particular, we're trying to create a score which treats every single independent experimental line of thought differently. So where I come from in Europe, we use the number 10 scoring things, and I'm sure that, I mean, the French use 20, but I don't know why the French use 20, but most people use the number 10, which I think is a great number. And so we decided that because there's 5 independent lines of experimental evidence, each one of them should have a similar scoring, up to 2, depending on how much intensity we have. And we have some rationale as to why you would want to treat them differently in different lines of a particular score. You want them to be independent when they contribute to the score, and we want the score to be interpretable. So we could do some machine learning, we could implement, for example, deep learning if we wanted to do absolute overkill, and just go in there and end up with a score which is a decimal, which is very hard to interpret. So, for example, even CAD scores, raw CAD scores, which are one of the most interpretable scores. When not scaled, they actually create really big problems for interpretation. So we decided to, using 5 different lines of experimental evidence, quantify each line. And so for eQTL information, any presence of an eQTL is already a good sign. So it's a 2 for eQTL information. Why? Well, because these eQTLs have passed quality control, and GTEx did a lot of work in quality control, and eQTLs did even more work. So any EQTL information is a 2. ABC contacts, they've also passed extensive QC, so they're also a 2. So if you have a SNP that only has EQTL and ABC, it has a 4 on the score because it has it has an EQTL presence and it has an ABC presence. So 2 different lines of experimental evidence. Motifs and K2 scores are kind of the same thing because they refer to transcription factor binding. And so each one of those scores is a 1. So if you have a motif, you have a 1. And if you have a K2 score, you also have 1. But you need to have both because you really need both to have good evidence of transcription factor binding. So if you have both, then you have 2 because transcription factor binding is an independent line of evidence. And so if you have EQTLs and ABC contacts and K2 scores and motifs, you have 6 because you have 2 and 2 and 2. And then the last 2 lines of experimental evidence are DNA accessibility. It's a very good predictive function. So any presence of DNA hotspots, DNA accessibility is a 2. And then finally, histomark information is also important, because if you don't have any histomark peaks in any of the roadmap issues for your SNP, it is very hard to believe that anything is happening. Unless you have a cell line which is outside of anything that roadmap analyzed, this wouldn't portray you possible, but is rare. So histomarks are also a 2. So we have some SNPs which have a 10, which means you have evidence across every layer. And those SNPs are very good to focus on, because, and typically they have high CAD scores, and they're typically some of the best candidate SNPs to look at. If you have a 9 is good. Okay, so don't think that a 9 or an 8 are bad. These are good scores. It just means that one of the lines of evidence isn't there, which may be had to do with function, and it may also be because you imagine if you're analyzing a melanoma GWAS data set. Well, melanoma, we have skin data sets, but we don't necessarily have every skin data set out there, and there's limitations in the original data. So just because you haven't got a 10, it doesn't mean it's not a good score. A bad score is a 2 or a 4, something like that. I mean, I think we're all familiar with scoring methods. But yeah, I mean, some things have a 0 because they have almost no data, and those are definitely very difficult to believe that they're actually doing anything, because they have no EQTL information, they have nothing. I mean, it's very hard to say, what is this SNP even doing? I don't want to say that maybe it's SNP with 0, which may be functional, but I just find it very hard to. I find it very hard to. I would find it very hard to believe. I mean, I can. Yeah, you can never say never. So yeah, gotta be cautious. So the FTA locus example, which is the example I'm showing here, we have evidence for EQTL associations and chromatin looping. So this is a perfect example of where long-range chromatin looping is driving a lot of these effects. And so the EQTL associations are over a megabase long, and the chromatin looping is also over a megabase long. And we also have TF motifs, we have DNA hotspots, we have histomarkers. So SNP is fantastic. The SNP is a very good SNP. And we didn't have K2 score information, so that's why it's a 9 and not a 10. Because the K2 score for this particular SNP, it wasn't identified, probably because there's fundamental limitations with the deep sequencing. K2 scores, we're trying to get more deep sequencing K2 information, because there's fundamental limitations in every data set. K2 scores are not, they're not, perfect. There's nothing that's perfect. It's all working.
Sorry, quick question. So, chromatin looping is usually non-allele specific. How do you make it allele specific?
When it disrupts local TF binding, and the TF is critical for the enhancer formation. Mm-hmm. So, chromatin looping, you're right, is not allele specific, but in this particular case, it's a weird example where it is disruptive. So, I think this is rare for SNPs, but in some SNPs, it does really affect transcription factor binding, and then the transcription factor isn't there, and then formation of the loop. I mean, I'm not going to say the loop is never there. We just have weaker interactions. So a lot of people say, oh, so this loops or this doesn't loop. That's not the truth. The truth is you either have stronger interactions or weaker interactions. And so I think the absence of that positive transcription factor does weaken the interaction of that for that. And it really affects expression of downstream genes. So so you're right. Looping is not allele specific, but alleles can affect intensity of contact. So if that's kind of real. That's a fundamental biological reality. A lot of people say it's a loop, and it's really just sporadic stochastic contact. So yeah. So this particular SNP is very good. It has a score of 9. We've sort of shown how we get the score here. And there's a lot of analysis on this to sort of evidence to provide evidence of function. It's pretty deep analysis here. And it's associated with white and beige adipocyte differentiation or proliferation. So it's a very, very interesting SNP because it helps the formation of beige fat, which is very thermogenic. So it's effectively a good way to lose weight. You can lose weight without even doing any sport, which is fantastic. Yeah, people love this allele. It's a small effect though, compared to if you do have a really bad diet, even if you have this SNP, it doesn't mean that that you will overcome that. So these things are complex and multifactorial. And I'm going to show one of the final SNPs that we're looking at here, COVID-19. So this SNP was associated with 40% reduction in risk of infection. So this is a massive effect. And so this particular SNP effectively has ABC interactions with ACE2. It's not really a mystery because effectively, if you look at the graph here, and I'm going to show you the particular graph for this particular variant. So can you see this really close to ACE2? So it's not actually surprising that it's interacting with ACE2. because it's just so close. So interactions are easier to have in SNPs. When you get further away, it's really hard to have interactions. This particular example is super odd. Okay, so this is like a long-range interaction, which is very rare. It's very rare to have such long-range interactions. So do not come out of this talk thinking that 1 megabase interactions are common. They're not common. But this is a very conserved regulatory element interactions. So that's yeah. Please go ahead.
Like we talk chromatic looping AEC interaction. So like this interaction need a basic structure, like gene in promoter and enhancer. At least need these 3.
Correct. So the interactions at a basic level are like this.
Yeah.
So enhancer promoter.
Yes.
But the reality is this, which is more complicated. So the reality is that you can have insulator factors that are also affecting 3-dimensional structure. So an enhancer and a promoter will contact, but why that particular enhancer, right?
I have the slides.
I have the slides, but feel free to take that. By the way, this image is fine. It's my own. I mean, you can share it. But the key thing here is why is that particular enhancer looping that promoter, right? And so we have, typically for promoter, we have more than one enhancer. We'll have several. So why is one contacting more frequently? And the answer is because of transcription factors, but also because of other elements that are constraining it. You're all familiar with TADs. TADs are basically large sections of chromosomes which interact with each other. And insulators can really affect that looping. And one of the key proteins for looping is CTCF. So it's a protein that mediates most loops. CTCF and cohesin are basically the basic scaffold which mediates a lot of DNA loops, especially short loops. So you need to have proteins to push it together, because if you just have DNA, it won't loop. So it's all mediated by CTCF, cohesin, and local TFs. Yeah, local TFs are critical. So the answer is you have a very complicated picture, but I mean, if you want to analyze many things, you start by just, enhancer promoter, which is going to be the majority of interactions. You also have promoter-promoter interactions. It's terribly confusing, but if the promoter is not that important for us, because we're interested in if your SNP is already in a promoter, then it probably affects its target gene more than any other gene. So it's enhancer SNPs that we're worried about, intergenic SNPs that we're interested in. So enhancer promoter. Yeah, I mean, forget everything that's on the other slide. Just enhancer promoter. That's the simple. So that's a straightforward way to see it. So that's the way that I see most of my analysis. So let's talk about the COVID-19 though. So evidence of 3-dimensional interactions. It's phrase 2. We have K2 scores, GNase hotspots. We have repeats, but we don't have EQTL information. And so it's interesting because this SNP does not have EQTL information, but has been shown experimentally to affect RNA expression when you actually do editing. So it's an interesting SNP. This is why I'm saying that a score of a 7, for example, is not a bad score. It's just it has to do with, we have to look at each layer of evidence and say, why is this missing? And maybe it's because it hasn't been analyzed. Sometimes people have done windows around promoters to analyze SNPs. This SNP is very close to the promoter, so it's definitely a simpler taxase too, but it just isn't available in GTEx data for some reason. Not all SNPs are available in all data sets, so that's another limitation. So this particular variant is very interesting. And let's do the web demonstration. So I'm going to this can always go south, so disclaimer. Let's make sure it's all live. Let's see. It works. So does anybody have any target SNP that they like? Any particular variants that they are working on right now? Any particular GWAS variant? Do you have the RS number that you can?
RS1 2020-03592.
You're going to have to repeat that. I hope it's on my database. If that's not on my database, then RS1
1220 3592.
Okay, I need to copy this. So here's your SNP, which has a score of 10. So that's fantastic.
We got a winner.
Well, okay, so let's look at your SNP. Let's look at your SNP in detail, because this method is here to be used. So, so this particular SNP seems to have massive signal for IRF4. Is this something that's consistent with what you know?
Yes.
Okay. So Okay, so let's have a look at location first. Okay, so this is why we have this browser shot. I don't know how fast this browser is going to work. But yeah, so it's basically overlapping an intron of IRF4, right? And it's a very big intron. Yeah, so I'm just trying to get a big picture here before UCSC sort of crashes on us. So yeah, so this particular variant is definitely in the intron of IRF4, the fourth intron of IRF4 for the main transcript. So that's good. That's the context. But let's go back to your variant. So the CAD score. So the CAD score is not super high, but that might be okay because this might be a regulatory variant, and a lot of regulatory variants don't have huge CAD scores because they don't affect survival because they're all enhancer affected. What phenotype is associated with this? Can you reveal that, or are you?
There is
You want to keep that confidential?
some like cancer related, lung cancer related, and bladder disease like that.
Fantastic. I have personally found that a lot of GWAS lung cancer, GWAS SNPs are very immune related. So this is my personal. When you do an analysis across FORGE2, the method I showed before, like you will get a lot of immune, sort of, for example, T cell associations. So that's, I mean, yeah, lung cancer has all these like immune associations, not in lungs. It's all in the immune cells. So this is pretty interesting. Yeah, so we have an association with IRF4 in blood. So that's pretty critical. From EQTL data. So the EQTL data is overwhelming. Can you see all this IRF4 sort of repeated and repeated again? That's a good sign that you have IRF4. I don't know if it's your favorite target gene, but it is a major EQTL target. It's also looping to IRF4. Can you see its ABC information? Yeah. So I'm going to explain every part of the web page because this is what a live level demonstration is going to be doing. So the first things that we have are closest gene, IRF4. We looked at the browser. We have CAD scores. The CAD score is not super high. So CAD scores you can see here is about 0.63, but it's not super low either. So a low CAD score will be 0.00 something. So this is an intermediate score. EQTL information. So the EQTL, we have EQTL information for IRF4 across many different tissues. You can see the tissues here. So lung, for example, is an EQTL in lung. It's also an EQTL in skin, in whole blood. In intestines, and EQTL across many tissues of T-cells. Okay. And actually, when we put EQTL in, this was also an EQTL of IRF4 in blood, and it's also an EQTL of XOC2, but we've only seen one XOC2 association, we've seen about 10 or more IRF4 associations. So, I mean, IRF4 seems to be important. Let's look at chromatin looping. So this is the activity by contact information, and here you can see it loops to IRF4 promoter. Okay. So this particular variant is a looping to an IRF4 promoter.
Does it tell you what tissue that looping was done in, or what cell type that looping was done in?
Correct. It tells you here, but the cell type is a bit cryptic. So it's in MM1S encode. So we need to have a look at what that particular cell type is. ABC did their experiments across many cell types because they wanted a lot of material. You need a lot of material for Hi-C. So it's not always like the cell type that you would wish. But yeah, we can have a look at this particular cell type, peripheral blood of multiple myeloma. So it's just a bloodline. Yeah, this is just a bloodline. So in blood, which I think is critical here because I've always seen lung cancer, the immune cells, like that's where the enrichments are.
Can you also scroll on the right? It says base overlap, something like this. Can you explain what it means? Base overlap 1, the last column.
Base overlap.
Yeah.
So that for almost all things it's going to be 1 because there's a single nucleotide polymorphism. So you may have an insertion, and then or you may have you may have a variation which is substitution or an inversion, and then you'll have like a bigger base overlap because we'll be dealing with a bigger region. It's important to have base overlap because you want to know how much overlap with the contacting segments you have. Because some Hi-C contacting segments are like 3 kilobases, and if you have an overlap of, for example, 200 bases, but it turns out that like you have an inversion which doesn't affect the whole region, then your Hi-C contact information is dependent on that 200 base pair overlap. So can you see what I'm saying? So the overlap is important because Hi-C is based on chunks of DNA that loop with each other, and the size of the chunk of DNA and your overlap with it matters. Yeah.
So How do we know if it's allele specific or allele enriched? Do you have any indication for that here?
So we give the alleles, we try to give the alleles in the QTL data. So I see the reference allele and alternative allele. For the EQTL data, we have the alleles. For the ABC, we do not, because ABC has never been looked at allele specific levels. So the ABC looping data is purely for a region, and it's usually very high level structure. So a SNP can't really, we've seen an example in FTO where really effective looping, but this is very rare. Okay, most SNPs do not affect looping. And so this is why we will not. So ABC and allele specific data, I mean, Jesse Enright, who's the guy who sort of invented ABC, I mean, it would be great to have allele specific information on ABC. I don't think we are at that level yet. Yeah, but that's like the future. So that would be a very interesting project. Yeah, we have Cato and FORGE2 TF information. So we have we have several different motifs overlapping. So we have one motif which has consensus overlap and a match of 8 times 10 to the minus 6 p-value. And we also have a Cato score for those particular alleles. So CNT affects binding in all of these different cell types. Can you see the cell types here? So the Cato score seems pretty consensus. And for some reason, the motif that the Cato score is identifying is for NFB. There's a TST1 motif in the motif match. These things are imperfect, but you want to see different motifs overlapping a site because it's usually a good indicator of regulatory element presence. Yeah. And then let's look at cell types. So obviously a huge amount of blood data. Can you see this enhancer mark in blood? So in B cells you have HVK4 mono, and in T cells you also have HVK4 monomethylation. So this variant, I would really analyze it in the blood cell line because this is all pointing to blood. So if you want to see enhancer activity, I would really look at blood. You can look at lung, but in the end, what will matter, I think, especially lung macrophages. But I mean, it's very hard to get lung macrophages. So what you want to do is probably just use macrophages and blood cell lines, which are the best proxy. Yeah. So this particular safety is a home run. So you're on a good path. Yeah, you clearly have a lot of data supporting your particular variants. So that's fantastic. And yeah, so I mean, this is a particular use case, but we're trying to expand it to as many variants as we can. We're currently 37 million variants, but there's always some variants that somebody just has and I'm not in on this, so we want to expand that. Yeah, and if anybody has any variants and wants sort of more support on particular variant annotations, we can do that. Yeah.
The system can predicate sleep on the virus is not human.
Sorry, can you?
Sleep on the HPV16 virus is not human.
Correct.
You can predicate this kinds of sleep don't have an RS number because they're not human.
Right. So a lot of symptoms don't have an RS number. So that's kind of a problem. But we can. So this we have a standalone version of FORGEdb that runs all those SNPs as well because we work on location as well. So we can work on SNPs that don't have an RS number. You were talking about the virus.
Virus, we found the HPV16 sleep in this specific region, but it's not a human being possible cannot input here.
Right. This is for human data. I mean, I think for non-human information, especially for viruses, you're mostly interested in protein structure. So yeah, I mean, obviously that's going to be by the way, a lot of you work on mouse as well, right? Or you're interested in you don't work on mouse models? I wouldn't say anything. I think that it would be. I don't know if it's a good idea. We're going to out on this idea, but we would like to get more mouse data for mouse variants. So not human variants, but just mouse variants. So sometimes some mouse variants and human variants like the similar regulatory elements. And I mean, this is rare, but we would like to get an expanded encyclopedia. So that's kind of something that I'm interested in. But the human version is already out there, and this is all publicly accessible. Online. So we definitely alive for the human data. Yeah, which is the most important data set right now. I thought those mouse enthusiasm, but it's too much. So, Yeah, so this is fantastic, and I didn't get your name, but
So my name is Jin Hu Yi.
Okay, well, this is fantastic. Your SNP seems to be one of the top SNPs that are in the list, so that's good. We have a distribution of SNPs in scores, so we have a more or less normal distribution around 5. So your SNP having a 10, that's pretty high up. Yeah, and we have a bunch of analyses showing the FORGEdb scores like really correlate with GWAS p-value scores, and also with MVARs. I don't know if any of you worked on MVARs. MVARs are massively parallel reporter assay data sets, and we've also validated the scores on those MVARs. So, I mean, scores are very simple conceptually, but we've done a lot of validation on it. So it's not, what we're trying to do is get something that's explainable. Yeah. So, so this is a method, and I'm glad that it didn't just work, but we actually got some interesting information for your variants. So, so thank you very much, everybody. This has been fantastic.
I think we have questions on the chat. Back to the closest gene, does the closest gene have a max distance allowance to the SNP?
Right. So on a chromosome, there will be a closest gene, right? Because every chromosome has a gene. There is currently not a distance because we're trying to keep the closest gene at any level. But I do think that you should look at the browser. We provide the browser score in part because we were scared that somebody would be like, oh, closest gene is like 3 megabits. What's closest gene? And, browsers, this is why it's important to look at the browser shots, because we will provide a closest gene for every step, because there's always a closest gene, but it might not be close. Right? So this is why, the browser shot is important to evaluate. Yeah. Be careful with be careful with just getting the closest gene and running with it, because it might not be close. Closest doesn't mean close. It just means the closest. Yeah. More questions?
And then someone also said, I wonder how the score would look if you add in the deactivating marks like H3K27 for methylation. It would be interesting to see if there are any positions having both active and repressive marks, and how would you account for that with the math?
So the reality is for every SNP out there, there's always an overlap with some active mark and also with some repressive mark in some tissue, right? So if you look at one variant, and so let's look at your variant which has a 10. Okay, this is like the best. You'll see it overlaps active and repressive marks. So you see it in this monocyte data set. For one of the monocyte data sets, it overlaps 2 marks. Why? Well, because you have overlapping histone marks. Okay. So it overlaps a repressive mark. And it also overlaps a transcription mark, but mostly, it overlaps a repressive mark. But then in another data set, for example, B cells, it overlaps an active mark, so H3K4 mono. So the reality is that for different cell lines and tissues, one cell line's repressed region is another cell line's active region. However, I think the question is also around bivalent regions. So you do have some regions which have H3K27 trimethylation and H3K4 trimethylation, and those are like bivalent promoters, a special type of bivalent promoter. And for those, it's very important to look at these overlaps. So I think that question is especially useful if you're interested in bivalent promoter regulation. Yeah. But the reality is that almost every enhancer in one cell line is somehow repressed in another. So this is why the cell type specific context is so important. And spoiler alert, we're developing a version of this method that you can select the cell line that you want, and we'll give you all the data. So currently it's static. I'm just giving all the information. But we're working with CBIT to effectively just get a version that you will select your cell line of interest, for example, lung or blood, and then it will just give you all the information for that cell line. Because cell line context is so important, right? If you just look across every cell, then in the end, it's a huge model of data, and this is why it's important to select your target cell line. So this becomes more readable. It's not the most readable web page in the world. So we need to collapse these tables and make them cell type specific. Yeah.
And maybe also to have a link that you can go and show the plot, especially if you have some contrast, the publication ready, so to say.
Exactly. Yeah, completely agree. So we're trying to provide more functionality in terms of interactive data sets. We provide links to all of the, not just the original data, but we want to provide links to, especially to the EQTL associations, so we can plot those, and also kind of have a plot for the score, which kind of describes your score, like a plot on a paragraph. So that's what I'm trying to work on some kind of score explanation algorithm for every single SNP to tell you why it has this score. Because I feel that just describing the score is not always clear in all these tables why it's being different. So we're working on making an interactive page. Yeah, currently this page is very static, so we're working on making it interactive. Yeah, and yeah, and I'm so glad that this must be like the best. I promise that I haven't talked to to anybody before the talk to give me a random snip. This is not a setup. This is not a setup. Okay, this was completely independent. There is no secret WhatsApp group where we were talking about before. Give me this variant. No, this is definitely this is definitely spontaneous. But I'm so glad that this is working and that that we can use this method to filter out the key snips and save time. Yeah, just go ahead.
So how are you planning to validate the score based on what is already known? Because so far it's a really, really, really nice resource that we can pilot all of these sources. But then when you have this score, what is it with the reality that this is already out there? Let's say like an internet does that thing, right?
Correct. So what we really want to look at is we've been looking at a lot of MPRA data to try to see MPRAs are. So MPRAs with luciferase assays have limitations. Okay, they have limitations, but they are one of the best sources for extensive sort of validation of particular data, particular SNPs. And so we've been looking at how our score works with MPRA data. Specifically, we've been looking at MVARs and other data sets. And so, I mean, essentially, you can reach a point, especially with the MPRAs, where you have. I'm just going to try to get you some data on the charts here. But for the MPRAs, let's see. Yeah, so this is a particular. Can you all see this graph? So FORGEdb scores are, especially the top scores, like a 9 and 10, they're strongly associated with MVAR proportion. So having like having functionality in the bigger SNP. This is a very preliminary analysis, by the way. So that's why I was sort of looking for this graph in files. But we do have good relationship with PRA. Yeah, and we've done a lot of other analysis outside of MVAR, looking at GWAS SNPs and especially looking at GWAS p-value, because GWAS p-value across multiple traits will give you an idea of how functional it is. So the answer is yes, but we're also working on expanding that. Yeah, and luciferase assays aren't perfect, but they are the best proxies. So it's all work in progress. Yeah, it's work in progress. Yeah, and thank you so much. I hope I'm not standing between anybody and lunch. It's already 2 o'clock. So yeah, thank you very much for your questions. Yeah,
and this is integrated into LD Link.
Exactly. So this is definitely. Yeah, so this is integrated. Every single FORGEdb score is integrated into LD Link. Every single FORGEdb page is part of LD Link. They're like brothers. So like we're trying to get them like integrated. Yeah, I'm really keen on also making graphs within LD Link and making them work. So yeah, Mitch and I are working on that. So yeah, that's what we're doing. Yeah. Anyway, yeah, thank you very much. Yeah. Any other questions? Cookie box. Yeah.
Yes.